So we're going to start the next session. It's our engineering vascular tissue session. Our first speaker, Dr. Yibing Qyang from Yale University.
I have pointer, but the speak phone. I can use this. Okay. Okay. Thank you. Okay. Thank you. Good morning, everyone. Thank you for coming, and it's a great pleasure to attend this seminar. And I really enjoy the symposium, and the science is great. The party and the music were excellent. So it's a great event. Hope you all enjoy it. So today I would like to share with you our progress about pluripotent stem cell for heart and blood vessel repair. And there's no disclosure. And I would like to share the islet 1 cardiovascular progenitor-based repair and human iPS tissue engineered blood vessel, and a couple progress slides about preclinical swine iPS modeling for engineering. And our group is interested, as many groups here, starting from patients with either cardiac or vascular disease, generate iPS cells, and either study disease in the dish to study mechanism for drug screening, or to generate a cardiac tissue or blood vessel tissue and for a cardiovascular repair. And so the heart failure affects more than 5 million people in the US, and 62% of the cases caused by myocardial infarction. And the cellular therapy could be a potential strategy to restore the loss of the myocardium. We start from a different perspective, utilizing developmental biology as our guidance and understanding what kind of cells form the natural heart in life. So starting from the wonderful developmental biology study from Dr. Sylvia Evans' group from UC San Diego. And so the islet 1 precursor cells, by lineage fate mapping, can generate the cardiomyocytes in a murine embryonic heart, and as well as adult heart. So the islet 1 is a limb homeodomain transcription factors, and it's expressed specifically in cardiac precursor cells and not in mature cardiomyocytes. And so in Dr. Kenny Chen's group at Harvard, I led the effort to discover canonical winged pathway, which is required for self-renew of islet 1 precursor cells. And the winged inhibition is required for the pre-specification and cardiovascular differentiation of these progenitor cells. And so the challenge is in adult human being hearts, adult animal hearts, the islet 1 precursor cells exist as a very rare population. So taking advantage of embryonic stem cell approach, we generate a mouse line in which the GFP is under the control of islet 1 and GATA4 sites. And starting from inner cell mass, we generate mouse ES cells. And then through mouse EB differentiation, we can purify GFP positive cardiac precursor cells for research as well as cell-based therapy. And so as alluded in many talks at this symposium, the survival of implanted cells is a huge challenge. So as we all know, heart cells like to exist in aggregate or spheroid form. So we hypothesize that generating islet 1 cardiac precursor cell spheroid may greatly enhance the survival. And we utilize the methylcellulose in low attachment 96-well plate. We can generate cardiac spheroid within 12 to 24 hours. And so it appears the spheroid formation can enhance the collagen and beta-1 integrin activity. And if we block integrin and collagen activity by RGD peptide and collagenase, the islet worm's fluid could not adhere to the dish in vitro. And we further extended this approach into human cardiac progenitor assay. And by Wnt activation and Wnt inhibition for human ESA, iPSA differentiation, we generate islet 1 NKX double positive cardiac precursor cells. And utilizing methylcellulose and LOX kinase inhibitor, we can generate human islet 1 cardiac precursor cells as fluid. And so since mouse cardiac fluid is independent of the LOX kinase inhibition, but the human's fluid formation requires LOX kinase, so we want to understand the potential mechanism about LOX inhibition and fluid formation. The LOX kinase has been implicated in inhibiting low RAC GTP, which is required for cell-cell adhesion or fluid formation. Indeed, we discovered that the LOX kinase inhibition can lead to activation of the RAC GTP. And if we inhibit the RAC activity, fluid is destroyed. And when we inhibit downstream mediator of RAC junk, fluid is also destroyed. But however, other MAP kinase inhibition does not affect fluid formation. And the beta-catenin and fluiding staining indicate that LOX inhibition can generate a nice fluid cell-cell adhesion. And the RAC GTP activation can lead to the IQ gap1 recruitment to the cell membrane, therefore stabilize cell-cell adhesion. And we perform the immunostaining and indicating that IQ gap1, the RAC, and fluiding, and so they have wonderful colocalization after we inhibit the LOX kinase activity. And we analyzed catecholamine activity, and the N-catecholamine is induced by fluid formation, and N-catecholamine is abundantly expressed in the cardiac progenitor cells. And the co-staining of catenin, catecholamine, and fluiding indicate a nice colocalization in fluid formation, but not in the control treatment. And so we next label human cardiac precursor cells with luciferase construct, thanks to Dr. Zhu's support. And so interestingly, the fluid formation can greatly enhance the survival of islet 1 precursor cells. So day 3 and 4 week luciferase signals were quite comparable. And we performed histology 4 weeks after fluid implantation. Human leukocyte A represent a human origin, so we observe that the human cardiac precursor cell can differentiate into cardiomyocytes labeled by troponin T. And these human precursor cells can form mature microvessels, and by the HLA staining, and also the presence of red blood cells in this microvessel. And human cardiac precursor cells can also form hybrid microvessel together with the mouse endothelial cells. And we performed the fraction shortening and ejection fraction, and human cardiac precursor cells can enhance the cardiac function. And the interesting thing is that this LOX kinase inhibition and methylcellulose can help all the cell types to form the organoid structure, starting from ES cells, pure endothelial cells, cardiomyocytes, or cardiomyocyte endothelial co-culture, even neuronal precursor cells, and functional mature GABAergic neurons, indicating that this approach is a general approach for organoid formation, for tissue repair and regeneration. So I would like to share the second avenue of our research, which is about vascular disease and tissue engineering. And we start from a patient with elastin mutation. In this patient, the ascending aorta and coronary artery are narrowed or blocked by the overproliferation of vascular smooth muscle cells. And we generate iPS cells from these patients and generate smooth muscle cells from iPS cells. And we discover that the patient smooth muscle cells, they have defective actin filament bundle formation, which is required for contractility. And this is consistent with the idea that patient smooth muscle cells, they lose contractility, they proliferate and migrate, and too much compared with control. And I will have a couple of slides to show this phenomenon. So besides the 2D Petri tissue study, we generate the iPS smooth muscle cell tissue ring by taking advantage of the self-assembly activity of smooth muscle cells. So iPS tissue ring can be formed and histology can be performed. And more importantly, we can measure the contractility through collaboration with our engineer and for this tissue ring. And interestingly, the patient tissue ring contracted less compared with healthy control, and the patient tissue ring proliferated much faster than healthy control, consistent with in vivo patient phenotype. This is our current working model, starting from elastin deficiency and finally toward smooth muscle cell hyperproliferation. We try to rescue the actin filament bundle formation, perform drug screening to treat the patient in the dish, and eventually to our humanized mouse elastin deficient model. And so taking advantage of unlimited supply of human iPS-derived blood vessels, so we want to ask the question whether we could generate autologous or allogeneic iPS blood vessel for patient repair and regeneration. And our wonderful collaborator and mentor, Dr. Laura Nicholson, has utilized human primary donor SMC for vessel repair and regeneration. And clinical phase 2, phase 3 trials are ongoing. And although very exciting, Laura and I, we discussed we want to move the field forward by utilizing the iPSS muscle cells to address 3 major challenges in the field. First, primary donor SMC, they have differences in collagen synthesis. You can see different age and sex. Those donor-derived SMC, so those kind of blood vessels, they have very variable biomechanical properties such as rupture pressure. So the second challenge is primary donor SMC, they have limited proliferation ability. And lastly, the dysfunction of vascular muscle cells in diseased patients may not remodel acellular graft very effectively. So that for those patients, they may need autologous or cell-based therapy. And so as a proof of principle, we generate iPSS smooth muscle-derived blood vessel. We implanted them into nude rat abdominal aorta and performed a 2-week study. And we discovered that there is a minor dilation after 2-week study, and we try to enhance this approach. And the HNE and collagen staining indicate cellularization and collagen formation, and macrophage staining indicated that there is active remodeling in the implanted vessel. Again, utilizing human HLA marker to label human cells, we see the human iPSS muscle cells, they express SMA, they express myosin heavy chain, indicating maturation in vivo after implantation. Now, by including cyclic stretch, we can further enhance the vessel mechanics. So in our previous reporting by materials, it's about 500 millimeter of millicurie. Now, utilizing a stretch, we can reach almost like 900. And its ongoing effort is try to further enhance the property. And lastly, a couple of slides about preclinical large animal modeling. For example, for autologous stem cell-based therapy from patient to the same patient, human to monkey model does not mimic this approach. So work with David Sachs at Mass General now in Columbia. We start from pigs, syngeneic or inbreed pigs. So one heart from one pig can be implanted to the other pig without any immune suppressor. So pigs are healthy. So using this concept, we generate 1 swine iPS cells. We can generate a cardiovascular construct that can be tested in all the swines. So therefore, utilizing this near autologous strategy, we can derive basic science that can guide patient-to-patient therapy. And we use doxycycline strategy, generate doxycycline-inducible swine inbreed iPS cells. In the presence of doxycycline, swine iPS cells maintain all the stem cell property. And so then during differentiation, we remove doxycycline. So therefore, transgenes will be off. And we observe nice vascular smooth muscle cell differentiation and labeled by smooth muscle cell marker actin and myosin heavy chain. And as a proof of principle, we generate vascular tissue utilizing PGA scaffold. And we label swine iPS cell SMC with GFP and culture for 10 days to generate vascular tissue, and then implant the tissue into swine utilizing subcutaneous model. We observe that swine iPS cell SMC can survive and by GFP staining, engraftment, and smooth muscle myosin heavy chain expression that we reported last December. So ongoing efforts is to further optimize this strategy so the community could utilize this approach for study in clinical model. So we, this is the first time to report the lineage tracing-based IL-1 precursor cells for cardiac repair. And this methylcellulose fluoride approach could be utilized as a general delivery approach for organ repair. And we generated the first implantable blood vessel product. And the inbreed preclinical swine model stem cell lines could be a promising preclinical large animal modeling for the community. Thank you for your attention.
We have time for one quick question. Yes.
Great talk. So do we know that Yamanaka factors? Do we know that Yamanaka factors can indeed reprogram pig cells into bona fide iPS cells?
Yeah. So this is a great question. So this has been a challenge for the field for more than 10 years. So in our current approach, when we remove doxycycline, the current swine iPS cells could not maintain pluripotency. And although they can differentiate into vascular smooth muscle cells very effectively, they could not differentiate into cardiomyocytes very effectively. We try to use unbiased small molecule drug screening to further optimize the swine pluripotency, because utilizing dox strategy, we can perform unbiased screening. And we are thinking about biomechanical differences between swine embryo and human and mouse embryo. So lots of basic science will be done in collaboration with Conrad Hochlingo and Sean Ching, the hardcore pluripotent stem cell experts in Boston as well as in New Haven. Yeah, thanks. So it's ongoing effort. Thank you.
Great. Our next speaker is Dr. Joe Berry from